Now, next is again another question with respect to your pollination. So, the 19th question: in which of the following both autogamy and geitonogamy are prevented? Okay, so the options given were maize, wheat, papaya, castor. This is a NEET 2020 question. Okay, so now let us see what is the difference between different types of autogamy. So, anybody knows what is three different types of pollination?
Autogamy, gnetogamy, and xenogamy.
Very good, excellent. Autogamy, xenogamy, and geitonogamy are the three important types of pollination. And pollination, we know that, of course, is a process by which you are going to transfer the pollen grains from the male and you are going to put it into the gynoecium of the female flower. So that is the pollination, and pollination is going to be three types. Okay, so just for your revision purpose, let us see what is self-pollination. Self-pollination, as you can guess from the word, it is nothing but movement of more pollen grains, which is the male gamete, from the androecium of the same flower to the gynoecium again to the same flower. So when the things are going to happen or the transfer is going to happen within the same flower, we call that as a self-pollination. Okay. Next one is nothing but geitonogamy. Geitonogamy is nothing but transfer of pollen grains from one flower to another flower, two different flowers, but it is going to be from the same plant. Okay. So in case of geitonogamy, the offspring produced, will it be genetically identical or it will be genetically dissimilar? Any idea?
Ma'am, can you repeat?
Suppose you have a fertilization happening by geitonogamy process. So at the end of your fertilization, you will produce seeds, right? The seeds that are produced by geitonogamy or the offsprings that are produced by
Ma'am.
yeah, very good, excellent. They are going to be genetically similar because they all are derived from the same plant. Because this plant is going to be genetically similar. Excellent, very good. Whereas in case of xenogamy, what happens is there is going to be transfer of this pollen grain from one plant to another. So they will be genetically similar or dissimilar?
Genetically dissimilar.
Genetically dissimilar. Okay, very good. Okay, so having all known about all these basics with respect to pollination, now let us have to remember a few important points regarding the plants. Okay, so papaya is a dioecious plant. So dioecious plant, what does dioecious plant mean?
They have separate sexes, no?
Yes. So there, suppose you have a male papaya plant, there is also a female papaya plant. Okay, so a male papaya plant will not have female reproductive part, that is, they don't have gynoecium. Similarly, a female papaya plant will not have androecium. Okay, so it is a dioecious. So please remember, whenever you see papaya at garden or whenever you read papaya, remember papaya is a dioecious plant. That is, you have a separate male reproductive part carrying plant and separate female reproductive part carrying plant. So papaya is a unique one which is going to be dioecious in nature, that is, it is going to have two different listings. Therefore, because they already have two different separate plants, the chance that the pollination will happen within the same flower or two different flowers of the same plant is going to be not there, right? You understood now? So the concept of pollination, self-pollination, that is whether it's going to be autogamy or it's going to be geitonogamy, is highly impossible in papaya because you have papaya plant independently carrying two different sexes. Whereas when you see the other one, castor and maize, they are going to be monoecious. Okay, monoecious means they have both the sexes present in the same plant. So if they are going to have the monoecious and unisexual, what is unisexual? Unisexual means they have both male and female. That is, androecium and gynoecium will be present in the same, the same flower. So what will happen if the both of them are present in the same flower?
Autogamy and geitonogamy is also possible.
Autogamy is possible, not geitonogamy. Why? Because in case of geitonogamy, here if you see they are bisexual. Bisexual means androecium will be present in separate flower, gynoecium is going to be present in different flower, but same plant. Bisexual plants means they have both the sexes present in two different flowers of the same plant. Therefore, wheat will actually undergo geitonogamy. Okay, you understood, ma?
Yes, ma'am.
So wheat can undergo geitonogamy, whereas castor and maize they can have both autogamy and geitonogamy both together. Okay, so please remember papaya will always is a dioecious plant, therefore they will always undergo only xenogamy. They cannot undergo autogamy and geitonogamy. Okay. Now next question is Ramachandran plot. So anybody knows what is Ramachandran plot? Yes.
No, ma'am.
Right. So actually, Ramachandran is a Nobel laureate. Okay, he got Nobel Prize and he is from India. Okay, so he actually invented or he made an important contribution in the field of molecular biology. What he did is he developed a plot that is called as Ramachandran plot. So in the picture that you see here is actually called as a Ramachandran plot. Okay, so whenever you have this MBBS course in the first year, you will have biochemistry. Okay, so biochemistry, the first fundamental topic that you will be having is going to be Ramachandran plot only. Okay, even in whenever you're going to write NEET PG also, definitely you will have questions coming from Ramachandran plot because it's a very groundbreaking research that has contributed by Indian. Okay, fine. So, like how we have Aryabhatta from India who contributed mathematics. Similarly, Ramachandran from India also contributed a lot to the field of biochemistry. Okay, right. So Ramachandran he developed a plot called as Ramachandran plot. And what is the function of this particular plot? The function is to understand the structure of proteins. Okay, so everything has its own structure, right? Hemoglobin has its own structure, myoglobin has its own structure. Okay, DNA has a double helical structure, RNA has a single standard structure, right? And triacylglycerides, of course, it's a lipid. Lipid has its own backbone structure, branch structure, or linear structure, right? So Ramachandran plot is actually a very important plot in order to understand the structure of proteins. Okay. So, do you know what are the different types of protein structures? How can
Alpha helix and beta plated sheets.
yeah, beta helix. That is the secondary structure, primary, quaternary. Any idea? Yes. Actually, when you talk about proteins, proteins have four important types of structures. Okay. The first structure is called as a primary structure. The second one is called as a secondary one. Third is called as tertiary, and fourth one is called as quaternary. Okay. These are the four important ways of classifying proteins based on the structure. Okay. So first, let us see what is the primary structure. Okay. So, what are proteins made up of? Oh, protein is a polymer of what? Anybody? Protein is actually a polymer of amino acids, right? Proteins are
Yes,
made up
ma'am.
of amino acids. And how many amino acids do we have? A cell will have 20. Okay, this number is very important. Okay, please remember a cell will have 20 different types of amino acids. Like you have serine, threonine, methionine, then phenylalanine, tryptophan, like that. So many 20 amino acids are present. Okay. So protein is nothing but it's a polymer of amino acids. And how about DNA? What is it polymer of?
Nucleic acid.
Nucleic acids. It's also called as a nucleotide, right? What are the three components of nucleotides?
Sugar, phosphate, and nitrogenous bases.
Excellent. Sugar, nitrogenous base, and phosphate group. Four different types of nitrogenous bases that are present in DNA.
Purines and pyrimidines.
Yeah, that is two. What are the four bases present in DNA? Let me mention DNA. Yeah. Adenine, guanine, thymine, and cytosine. Right? Do you have uracil in DNA?
No, ma'am, it's in RNA.
Excellent, very good. Okay, so DNA and RNA are nothing but polymers of nucleotides, and proteins are going to be polymers of
Amino acid.
amino acids. Very good. Okay, so it's a polymer. That is, many amino acids are going to be joined. And what bond is present in case of proteins? How are they joined? There's a special bond, right, which helps this particular amino acids in a protein. They are called as a peptide bond, right? Peptide bond is a CONH bond. Okay, so peptide is a bond that attaches one amino acid with a subsequent amino acid. Okay, right. So having known that the peptide bond is an important bond which connects the amino acids, there are, as I said, there are four different types of protein structures. First one is primary structure. So primary structure is nothing but a linear chain of amino acids linked together by peptide bond. The second one is nothing but the secondary structure. So what are the different types of secondary structures in protein? We all know
alpha
that
helix,
alpha helix, then
and beta sheets.
beta sheets, and there is one more which is called as random coils. Okay, means they don't have any proper structure, they're just loops. Okay, that's called as a random coil or this thing. So Ramachandran plot, in order to understand the structure of proteins, he developed a plot that is called as Ramachandran plot. Most important, you have to remember the x-axis and y-axis for a Ramachandran plot. Very important. Okay, so in the picture you see on the left side you see this psi. Psi is nothing but psi. Okay, so this is the Greek symbol psi. Okay, it's a plot of psi versus phi. Okay, so phi versus psi, that is psi versus phi, is a plot, okay, which is important to analyze the secondary structures of proteins, which includes your alpha helix and beta sheet. Okay, so for your level, for this particular NEET UG level, this much information is enough. Okay, later on, you will, whenever you do your medicine, there you will be actually given graphs. Okay, so everything has a meaning. So the yellow chart that you see here, the red bar that you see here, everything has a meaning. It's nothing but it's an angle, bond angles. Okay, so that is little advanced version. So for now, you have to remember that Ramachandran plot is important to analyze the protein structures, and it's a plot of phi angle versus psi angle. Okay, these two angles are present in the peptide bond which connects the amino acids of a protein. Is it clear? This much.
Yes.
Next is a little easy question. Okay, so which of the following is not a feature of active transport of solutes in plant? Okay, the options given are it requires ATP. What is the full form for ATP?
Adenosine triphosphate.
Adenosine triphosphate. Okay, so the options given is you have to actually find the incorrect statement. Okay, right. So it requires ATP against the concentration gradient, non-selective, occurs through membranes. So anybody, what is the answer? It's of course displayed. The answer is non-selective. Okay, so let us figure out what is the reason. Okay, so active transport is nothing but it's an energy consuming process. Okay, if you talk about transport in cell, there are three important types. One is a simple diffusion, the second one is facilitated diffusion, and third one is active process, right? Both in simple diffusion as well as in facilitated diffusion, it is a non-energy consuming process. That is, you're going to move molecules from high concentration to the low concentration. So here, if you see on the top side, you have more molecules, and on the bottom side, you have less molecules. Therefore, by nature, because of the difference in concentration, these small solute molecules are going to move from top to bottom. Okay, so this is nothing but simple diffusion and facilitated diffusion. But in case of active transport, you see the reverse, right? You see more molecules are present on the top side and very less molecule is present on the bottom side. That is, in case of active transport, you're going to move molecules from low concentration to high concentration. Because you're doing a process which is against the nature, you require energy in order to drive this particular process. So energy in case of active transport is going to happen by molecule, which is the ATP. So do you know what is the energy value for one molecule of ATP? This is also there. From one molecule of ATP, how much energy do you derive? Okay, so when you break one molecule of ATP, you actually derive 30.5 kilocalories per mole. Okay, so this value you have to remember. That also was asked in some year back NEET question. So from one molecule, I actually derive 30.5 kilocalorie per mole. Fine. So in case of active transport, you require energy in the form of ATP molecules to move, the solutes from low concentration to high concentration. And if you see this process, okay, all the three processes is going to happen via a membrane. What membrane is present in cells? What do you call?
Semi-permeable membrane.
Semi-permeable membrane. But in cell, what do you actually call this membrane as?
Plasma membrane.
Plasma membrane. Plasma membrane is also called.
Sorry, ma'am.
Plasma membrane is also called as, what is the other name for plasma membrane? It's called a cell membrane. Okay. It is also called as plasma lemma. Okay. So please remember plasma lemma. And who proposed the structure of plasma membrane? Any idea who proposed the structure of plasma membrane?
Is that fluid mosaic model?
Singer and Nicholson. Okay, so Singer and Nicholson again they got Nobel Prize for discovering the structure of cell membrane. It's going to be made up of useful lipid
Fluid mosaic
bilayer.
model.
What is that one?
Fluid mosaic model.
Yes, very good. Liquid mosaic model. Where is the what are the two types of proteins present in the in case of your cell membrane? There are two important types of proteins present. What are they in cell membrane? Integral proteins and peripheral proteins, right?
Yes, ma'am.
What is an integral protein? An integral protein, as you can understand from the word, integral means it is integrated into the membrane. So the green, actually the green picture that you see in the figure, it is actually a channel, and this channel is actually made up of integrated proteins. Okay. If you have proteins floating on the top, what do you call that as? Membranes also have proteins floating on the top. So what do you call those proteins as?
Cell protein.
Proteins. Okay, so please remember there are two types of proteins present. The peripheral proteins are just floating on the top, whereas integral proteins play a very important role because they act as a channel. Okay, this one, the green ones that you see here is actually made up of proteins, and they help in movement of molecules across the membrane because we know that these are hydrocarbons, right? It has a hydrocarbon lipid layer. Hydrocarbon will repel the water molecules. Then how do you actually put water inside the cell? Do you know what is the protein which helps in transferring water molecule inside the cell? There is a special name for it. That is also a neat question. Any idea? They are called as aquaporins. Okay, so aquaporins is it's a neat question. Okay, so aquaporin is actually a channel, or it is this kind of a protein which helps in moving the water molecules from outside to inside. Otherwise what will happen? We need cells need water to survive, otherwise it will die. Cells will die. If cells die, we all will die. Okay, so in order to transfer the water molecules across the membrane, because the membrane is having hydrocarbon chains, it will repel. So in order to transfer water molecules, there are specialized proteins called as aquaporins, which help in transferring the water molecules inside the cell. And it's an active transport because you need a lot of water inside the cell. Otherwise, the cell will undergo shrinkage. Okay. So again, that's about active transport. So here, if you see the question, the incorrect one is it's a non-selective process. Okay. Please remember there are three important features of active transport that active transport is a highly selective process. It's a very important point you should remember. Okay, so active transport is a highly selective process and it requires energy in the form of ATP molecule. That's the second point to remember. And third point to remember is active transport always happens, against the concentration gradient. So these three are important points that you should remember about active transport. So is it clear? Any doubts?
Yes, ma'am.
So what is the channel that transports water molecule inside the cell?
Aqua protein.
Aquaporins. Very good, excellent. So now next, let us go to a theoretical question. That which of the following bacteria reduce nitrate in the soil into nitrogen? Okay. So the options are Nitrosomonas, Nitrobacter, Nitrococcus, and Theobacillus. Okay. So what is the process when you actually convert nitrate into nitrogen called as? Okay, so let us start from the basic. Okay, so what is biological nitrogen fixation? Anybody? What is nitrogen fixation? Okay, so let me answer this question. So we know that our atmosphere has around 70% nitrogen, right? But the nitrogen that's just present in the air is of no use. Okay, cannot be used by plants. Therefore, in order to make those nitrogen useful for the plants, what there is a process is called as biological nitrogen fixation. Okay, so biological nitrogen fixation is a process by which you convert atmospheric nitrogen, that is N2 is getting converted into nitrate. Okay, that is NO3 2 minus. Okay, these nitrates will be present in the soil and this will be taken up by the roots and it will be supplied to the plant for the overall growth and development. Okay, so converting atmospheric nitrogen into nitrate is called as nitrogen fixation. Okay, So when you have this nitrate converted back to nitrogen, that is, you are going to convert NO3 2 minus into N2. When just convert, when we are just going to convert it back to atmospheric nitrogen, it is called as denitrification. Okay, so they are asking which of the bacteria is playing a vital role in the process of denitrification. So denitrification is performed by two important bacteria present in the soil. You have to remember the names, very important. They are Pseudomonas denitrificans and Theobacillus denitrificans. So the names are actually displayed. You can note down for your reference. Okay. So Pseudomonas denitrificans and Theobacillus denitrificans are the two important species of bacteria which is important for converting nitrate which is present in the soil back to nitrogen, that is NO3 2 minus getting converted into N2. Okay, so what is the process of converting nitrate into nitrogen called as
Denitrification.
denitrification? And what are the two important strains of bacteria that performs this process?
Pseudomonas and
Pseudomonas
Thiobacillus.
denitrificans. Okay, and what is the other one?
Thiobacillus.
Bacillus. So what is the right option for this?
Option 4.
Okay, yeah. Now, one more thing that you have to remember. So, for denitrification, it is Pseudomonas and Thiobacillus. For nitrification, so if you give if you see in the exam that it's going to be nitrification process, there are two important bacteria. They are going to be Nitrosomonas and Nitrobacter. Okay, so these two are exactly reverse process. Okay, so nitrification is facilitated by Nitrosomonas and Nitrobacter, whereas your denitrification is actually facilitated by Pseudomonas and Thiobacillus. So please remember. Okay, now tell me what are the important bacteria for nitrification?
Nitromonas and Nitrobacter.
Nitrobacter. So option 1 and 2 is for nitrification, and option 4, Thiobacillus, is actually for this thing. So nitrococcus as such does not have any role in denitrification or nitrification. It's just an odd man out. Okay. Right. So this is another technical interesting question. Okay. What will be the direction of flow of water when a plant cell is placed in a hypotonic solution? Okay. So if you remember, there are three different types of solutions possible. There are isotonic solution, hypertonic solution, and hypertonic solution. Okay. Hypertonic, isotonic, and hypotonic solution. Okay. So now let us see what is the isotonic solution. So isotonic solution is that particular condition where you have both the concentration of the solute both outside and inside the plant cell are going to be equal. That's called as an isotonic solution. Hypertonic solution is that particular situation where you have more amount of solute present, inside and less amount of solute present outside. Therefore, in order to maintain the equilibrium, what will happen is the water molecules will migrate from inside and it will reach the from outside and it will reach inside of the cell. So that's called as a hypertonic solution. Now let's see what is a hypertonic solution. Hypertonic solution is that type of solution where you have more amount of solute present outside the plant cell and you have very less amount of solute present inside. Therefore, in order to maintain the homeostasis or balance, you have to migrate the water molecules from the plant cell and you have to put it outside. So, in a hypertonic solution, what will happen is the plasma, okay, or the cells will actually undergo lysis. Okay, so this state is called as the plasmolyzed state. Okay, so hypertonic in hypertonic solution, the plant cells will become plasmolyzed. In isotonic solution, you have your the plant cells which will become flaccid, and in case of hypertonic solution, they will become turgid. Okay, so hypertonic plasmolyzed condition. In isotonic situation, you have a flaccid cell, and in hypertonic solution, you will have a turgid cell. Okay, so here, if you look at the questions, what will be the direction of the flow? The options are no flow of water in any direction. Option 2 is water will flow in both the direction. Option 3 is water will flow out of the cell. And option 4 is water will flow into the cell. So these are the four options. So if you see, the right answer for this particular question is option 4, that is water will flow into the cell. Okay, so the reason behind this, whenever a plant cell is placed into a hypertonic solution, water will flow into the cell because you have high water potential outside and you have very low water potential present inside the cell. Is it clear? Right. Now, next, let us move on to the 25th question. That is, which is the respiratory electron transport system located in the plants? Okay. So, there are four interesting options. The first one is intermembrane space. Option 2 is mitochondrial matrix. Option 4 is outer mitochondrial membrane. And option 4 is inner mitochondrial membrane. Okay. So, you know that mitochondria is also called as a powerhouse of the cell, which has two important, membranes. One is outer membrane, other one is the inner membrane. Okay, so electron transport system usually happens in the inner mitochondrial membrane. Okay, so remember. In the inner mitochondrial membrane that you can actually see in the picture, the electron transport system, which is also called as the ETS, will happen. Okay, so the right answer to this particular question is ETS will happen in the inner mitochondrial membrane. Right. Now, next one is this is a theoretical question. It's a NEET 2019 question. Which of the following is against the rule of ICBN? Okay, that is the International Code for Botanical Nomenclature. Is the full form for ICBN. Okay, so which is the following against the rules of ICBN is the question. So you have to find out the incorrect question, incorrect options given postulated by your ICBN. So the first one is a generic and specific name should be written starting with small letters. Okay, so we know that both the generic name and the species name of an organism should always be mentioned in the capital letters. So the incorrect answer is going to be option 1. So whenever you have handwritten, whenever you write the botanical names in the board, you can actually underline it. Okay, and every species should have a unique generic name as well as a species name. Okay, then only you can actually complete an organism's name. Okay, therefore the option 3 is correct. Option 4, it states that the specific names are in Latin and it should be utilized. Of course, the botanical names are always mentioned in Latin and it will be in Greek. Okay, so therefore the right answer, that is the incorrect odd man out among all these ICBN rules, is generic and specific names should be handwritten, should be written with small letters is the incorrect statement. Okay. Now next one is in Hatch-Slack pathway, the primary carbon dioxide acceptor is options are rubisco, oxaloacetic acid, the phosphoglyceric acid, and phosphoenol pyruvate. So these are the four options which were given. So if you remember in the Hatch-Slack pathway, the primary component for accepting the carbon dioxide is going to be phosphoenol pyruvate. So the right option to this is phosphoenol pyruvate. So the answer is displayed as phosphoenol pyruvate. Okay, right. Now, next important question that we'll be discussing in today's session is mad cow disease in cattle is caused by which of the organisms? Okay, it's usually mad cow disease is a disease. It's a prion-based disease that is present in case of cows. Okay, so it's due to a protein misfolding disease. Okay, so the options given for this particular question is option A, free DNA without protein coat. Then option 2 is inert crystalline structure. Option 3 is abnormally folded protein, and option 4 is free DNA without protein coat. Okay, right. So please remember that this particular disease, that is a mad cow disease, is actually caused due to prions. Okay, there are different types of diseases, right? Some are caused by virions, which we call it as virioids, and some of them are going to be caused by prions, which we call it as protein misfolding disease. So this particular mad cow disease is nothing but it's a prion based disease, and it is because of abnormally folding of proteins. Okay, right. Okay, so now next thing that you have to remember is okay, somebody else is also sharing the screen. So please remember, right? So we are talking about mad cow disease. So mad cow disease is actually a disease present in case of cows. Okay, and this particular disease is actually caused due to misfolding of proteins. Okay, so the right option is option 3, which is abnormally folded proteins. Is it clear? Right. Now, next question is which of the following statements is correct? Okay. So, option 1 is lichens are not good pollution indicators. Option 2 is lichens do grow in polluted areas. Option 3 is algal component of lichens is called microbiome. And option 4 is fungal component of leaching is called as the phycobiota. So you have to identify the correct option. Okay. So if you remember, lichens are very very excellent indicators of pollution. In the picture, you see that in the normal region, you see that there are dense growth of this particular lichens. But whenever you have polluted and dusty areas because of the smoke and nitrous oxide present, the lichens growth will be drastically affected. Therefore, lichens are very very commonly used as an indicator of pollution. Okay. So as you can understand, the right option is going to be option 2, which is lichens do not grow in polluted areas. Okay. Here, when you see algal component of lichen is called as a mycobiont. Okay, so that is not the correct one because algae doesn't combine with lichens. And similarly, the fungus does not, have a phycobiontic relationship with the lichens. Therefore, that's also an incorrect statement. The right option is lichens are always pollution indicators, and they do not grow in the polluted area. So the right option is option 2. Right. Now, next one is we have to identify the correct matches. Okay. So column 1 lists the different types of bacteria, and column 2 is actually listing the different situations where these bacteria can live. Okay, so if you can actually see this particular left panel, it is more or less it is to do with the archaebacteria. Archaebacteria are those bacteria which can live in extreme conditions of high salt, high pH, etc. Okay, and the left panel is actually different conditions, right? Hot springs, aquatic environment, guts, ruminants, salty areas, etc. Okay, so let us try to figure out what are the right options. Okay, so the first one is halophiles. So the word halogen, we know that it's going to be rich in high salt and all. Okay, so halophiles are most commonly seen in guts of ruminants. Guts of ruminants means if you see cattle like cow and all, you see that they have halophiles, and cow is very very important for producing biogas. So biogas produce பண்றதுக்காக we have halophiles, and halophiles வந்து எங்க இருக்கும்னா it is present in the guts of ruminants. So the option A will have um, fourth one. Next one is thermoacidophiles. Okay, so thermoacidophiles, if you see, is going to be option 1, that is hot springs. So thermoacidophiles. So thermo means what? Temperature. Okay, so where these particular thermoacidophiles can actually live in high regions of high temperatures. So hot spring is the right answer for thermoacidophiles. Methanogens, if you see, they are present in the guts of ruminants. And option D is that is cyanobacteria. Cyanobacteria are most commonly seen in aquatic environment. Okay, so if you match this, option 2 is going to be correct. That is A, which is halophiles for salty areas. B, which is thermoacidophiles for hot springs. Then C, methanogens for your guts of your ruminants. And option D, which is cyanobacteria present in the aquatic environment. Okay, so this is actually the right. And here I have mentioned the different logical reasons for mapping this particular bacteria for their different environmental situations. Right. Now, next question is, in Dicot Root, the vascular cambium originates from. Okay, the different options are intrafascicular and interfascicular tissue in a ring, then tissue associated below the phloem bundles and a portion of pericycle tissue above the protoxylem. And option 3 is cortical region. Option 4 is parenchyma between endodermis and pericycle. Okay, so if you remember, vascular cambium is actually the secondary meristematic tissue. Okay, therefore the right answer to this particular question is tissue located below the phloem bundles and a portion of pericycle tissue above protoxylem. Okay, usually vascular cambium is present in case of a dicotyledon plant. Right. Now, next one is which of the following shows mold phyllotaxy? Okay, so there are 4 options given. We are Calotropis, mustard, China rose, and Alstonia. Okay, so these are the 4 options that are given. So we know that phyllotaxy is nothing but it's a system of classification of leaves. Okay, there are 5 different types of classifying leaves based on the arrangement. One is alternate form, opposite form, then you have opposite Dicot state, and then you have ternate forms and cooled forms. So cooled form is again a 360-degree format where you have leaves rotating all the way around. Okay, so if you talk about old phalotaxy, old phalotaxy is actually seen in a plant which is called as Alstonia. Okay, so please remember this concept that in case of Alstonia, you will be having a old phalotaxy. Now, next question is regeneration of the damaged growing grass following grazing is largely due to options are secondary meristem, lateral meristem, apical meristem, and intercalary meristem. So usually, in case of grasses, you will have this particular intercalary meristem. So the picture shows this particular intercalary meristems. Intercalary meristems are frequently observed only in case of grasses, and they will help them to regenerate. Regeneration is nothing but its ability to grow back the organs again. So ஒரு organ damage ஆயிடுச்சுன்னா திருப்பி அந்த organ regenerate பண்றதுக்கான process is called as regeneration. Okay, for example, you see in lizard also whenever the tail falls off, the new tails emerges. It's because of the event which is called as regeneration. Okay, it's also called as the regenerative capacity of an organism. Okay, so intercalary meristem or regenerative capacity ஆலதான் even if the grasses are going to be grazed off by herbivores like a cow, goat, etc., it will be able to develop again and again. Okay, so the right answer for this particular question of regeneration of the damaged grasses is actually intercalary meristem. Okay. So, next question that we'll be discussing is bicarpellary ovary with obliquely spaced septum is seen in which of the following plants? They are Sebania, Brassica, Alu, and Solanum. So the bicarpellary ovary condition is actually observed in Solanum. So all sorts of the Solanaceae family, like your potato and all, they will exhibit bicarpellary. Next question that we are going to discuss is select the incorrect statement regarding inbreeding. The first one is inbreeding depression cannot be overcome by outcrossing. Inbreeding helps in elimination of deleterious alleles from the population. Next one is inbreeding is necessary to evolve a pure line in any animal, and continued inbreeding reduces fertility and leads to inbreeding depression. Okay, so if you see this, the right answer for this particular question is inbreeding depression cannot be overcome by outcrossing. So what is inbreeding depression? Inbreeding depression is that particular situation when we are going to have frequently crossing to the same generation. Okay, what will happen is this will lead to a development of an offspring which is going to be homozygous recessive. That is, the dominant characteristics is going to be lost. Okay, so it's a very negative impact on breeding process. Therefore, the right option to this particular question is inbreeding depression cannot be overcome by outcrossing. Okay, so I think with this we can wind up today's session. So if you guys have any doubts, we can work on it. Otherwise, we can continue in the next class.